Before Alexis, she's always like, Caitlin Jr., Kerrigan, I think she's class rate. I don't know what happened. Pretty early. Some of these kids are getting really good.
You want to just keep those, and if you see them come in, we can.
Sure.
Or maybe you can stay up here while we talk about the.
Yeah. Alexis.
Okay, so as you can see, the class overall did quite well on the second exam with a mean of almost 81%. And I was particularly impressed by the number of grades in the 80s and 90s on the exam. So basically, keep up the good work. If you're still struggling with this, then you really need to come and talk to me. You need to talk to Austin. We need to get this turned around. The point total that's on the front of your exam represents your current point total in the course right now. Obviously, it won't have the lab quiz on, which you're taking this week. But this really is about halfway through the course in terms of number of points. It does not include any optional points, okay? Optional points are calculated at the very end of the class because they change and the impact on your grade changes depending upon what your grade is, how many points you get, etc. So I calculated that at the very end. Remember, there's a cap of 4% on optional points that you can raise your grade by that particular amount. There was a question. On everybody's exam, I added 2 points. The reason I added 2 points was question number 28, which a lot of people missed. This question said, which one of the following is false about the electron transport chain? This is a true statement. It does contain proteins that are called cytochromes. It does require oxygen to function. It's found in the inner membrane. It does not pump H plus ions into the mitochondrial matrix. The electron transport chain pumps H plus ions out here. Now, what I think some of you were thinking about was the flow of the H plus through the membrane down the concentration gradient, but that isn't pumping. So the correct answer was D, but so many people missed that. That's why I added 2 points on, okay? If you got bonus questions correct on the last page, that was just figured into your total exam score. So your first exam score would have included any bonus points, and then I just added 2 points to everybody's exam after the fact because of that particular question. So any questions about the exam? Okay, did you all get the answer key that I sent you? I sent something this morning. I sent the PowerPoint slides for the yeast transport experiment, and I think they went through. Has anybody seen them on your email? Did they come through? I was worried it was going to be too big of a file, but there were only a few slides. So that will be a good thing to review for the lab quiz. Okay. This week in lab, what we're going to do is we're going to take the lab quiz first. You've got your study guide. You know what you're responsible for. The other thing then that we're going to do is I will turn it over to Dr. Morgan for a second if he wants to say a few words.
Yeah, as soon as you get to lab, you can start the lab quiz. It won't take... Well, there are a couple of questions in the very beginning that I have to do with the entire class, but you can start questions after that. It shouldn't take more than a half hour. After the lab quiz, we'll do a little bit of an overview of the challenges that you have this week in lab. We won't be doing any experimentation. We will be taking all those enzyme activity curves that you generated last week, and we'll be averaging the triplicates that you did for comparison. And then we will also talk about, well, how do you analyze it? What kind of information can you derive from these curves? All right? We have done one thing in the past that you can use, and that is to do a best fit line over a certain period of time. It has slope, and slope is the rate of the chemical reaction. But what about statistical analysis? How do you compare your results statistically? Talk about how to go about doing that. We'll also talk about the PowerPoint presentations that you will be giving the week you come back from break. For those of you who have lab on Monday and Tuesday, what this pretty much means is you got to have your PowerPoint presentation done before you leave this week, right? Unless you're especially adept at throwing something together at the end. But keep in mind that this is 100 points.
No. I'll have to look and see what it is on it.
It's not an insignificant part of your total grade, right? So be on time. If you can be a little early, that's great. There is a page in the particular lab exercise that I recommend you take a look at. It's page 4. It says Enzyme Project Group Presentation, so that you know what you are responsible for when you come back. All right? We'll talk some more about this beginning of the lab.
Yeah, there's a reference, I think, in the lab material at some point in time to doing a poster in addition to this PowerPoint presentation. We're not doing that, okay? That kind of didn't get taken out when it should have gotten taken out. So this week you'll be able to work on your presentation, hopefully get it wrapped up, and then when you come back you will be doing the presentation. And there's a lab we'll be working on too, isn't there, at the same time? Or
No, that's all we do.
that's all we do. We just do the presentation when we get back, okay. So any questions about your lab quiz? Even thought about your lab quiz yet?
If you've gone over the study guide, you will do just fine. The study guide is really well thorough as to what you're expected to know. And I think Dr. Browder mentioned before, go back to the material that you handed in after various exercises, go over that, all right, because you may see some questions similar to what was on there again. When we did the microscopy lab and you had the various types of single-cell prokaryotes, single-cell eukaryotes, heterotrophs, autotrophs, and so on, I told you at the time you will be responsible for identifying these organisms and knowing, are they autotrophs, are they heterotrophs, and so on. So be sure you go over that. You should have made some drawings of that during the lab.
There are some files on the computers in the lab that have those images, correct? I think they're on Cell Bio Resources.
Yes, Cell Bio Resources.
Yeah. But make sure you go through that yeast transport experiment and can follow the logic of, based upon these particular color changes or colors of solutions or colors of cells, what kind of conclusions you can draw from that information, because there will be a question about that. There will be a modified experiment where you do the same basic type of experiment and the same principles will hold, in that sodium carbonate's a base, acetic acid's an acid. The neutral red color changes to an orange yellowish color at the basic pH, a red color at the acidic pH. But there will be different outcomes than you got. And then you will have to answer those same kinds of questions. Does the data indicate that neutral red entered the cell? Does the data indicate that sodium carbonate entered the cell? So you have to understand the logic and how those particular results we got led to the conclusions we got. And if you understand that, then if you get new results, you can draw the proper conclusions from that. So go through those slides I just sent you, because it takes you through step by step and basically says, at this point you can make this conclusion. Understand why that's the case. Okay? So since we don't have a quiz today, since you don't have any questions about the lab quiz, unless there's any last minute, more than willing to answer questions. I'm not going to give up a class period, so we're going to start some new material. Okay? Make sure you have no lab questions. You all know how to understand, how to determine significant differences and determine degrees of freedom and look at a t-table and see what your 95% confidence level, what your 99% confidence level is. All the things you've been doing in lab are the kind of things you're going to be asked to do on the lab quiz. So it's nothing new. It's stuff you should be comfortable with already. Yeah, Macy.
What do we need to know about the intestinal goblet cells and the intestinal columnar epithelium?
You have to be able to identify which are which on a slide. Okay?
And just to clarify how we get our degrees of freedom for like a 95 and 99% confidence level so we can compare accurately. How do you, I just want to agree with that.
Okay, so anytime you do a t-test, you're comparing two data sets to one another. And so there will be a certain n value for each data set, which represents the number of measurements you take. In our case, it's the number of groups, okay? So degrees of freedom for a t-test is n-1 plus n-1. And then that number is, then you look at that particular row, and then you go over to the 95 or 99% column and find that value. And that is your expected t-value. Your calculated t-value, which you see in that little comparative matrix, has to be equal to or greater than that expected value for the difference to be significant.
For the RF values, that's like the amino acids, it's the distance it traveled, and then what else did it ever mean?
The distance the solvent moved, the solvent front. And if your solvent front is straight, then that particular value will be the same for all of those. Okay? A couple of questions, good. Anything else? Not lab quiz. Okay. So the next topic we're going to talk about is another aspect of metabolism. We're going to be talking about photosynthesis. And I have made some references to photosynthesis as we were talking about metabolism. But now it's time to dig into it in a little bit more detail. So this is one of those kind of outline slides that I like to throw in there to give you an idea of where we're going. So we'll talk about things like light-dependent reactions, light-independent reactions. One of the things that I'm going to do is there is a section at the end of the chapter which talks about alternative mechanisms of carbon fixation. That's something that I think is important, but we're just kind of running out of time to get into that in detail. So what I'm going to do is I'm going to make some questions on the next exam, which are bonus questions, which will deal with this particular section of the chapter. So it's about a 2 or 3 page section of the chapter. So that will be the bonus on the next exam, will be those particular type of questions. So you can prepare for the bonus if you want to by reading that section of the chapter and working through it. I made reference to the fact that when we talked about energetics, that photosynthesis is a critically important process for life on this particular planet. Because in this particular process, a unique group of organisms that we call autotrophs are able to grab this sunlight energy that is pouring out of the sun. Remember, there are these nuclear fusion reactions occurring in the sun, and that some of the mass of the sun, some of the hydrogen, is being converted into energy of several different forms. We call the totality of that energy the electromagnetic spectrum. And it includes things like damaging, gamma rays and X-rays, somewhat damaging ultraviolet energy, visible light, infrared radiation, radio waves. There's a whole spectrum of forms of energy that are being shot out into space and are hitting our planet. And photosynthesis depends upon the part of the spectrum, kind of in the middle, kind of in the sweet spot, where there is still enough energy associated with that particular form of radiation to do some work, but it's not so much energy that it's damaging the cells. We're in that kind of Goldilocks zone. Here in this rainbow, you can see that the visible light is actually a spectrum of several different colors. Shorter wavelength blue and violet forms of radiation, and longer wavelength red forms of radiation. And so this is the energy source that these organisms, these autotrophs, are harnessing and turning into the chemical energy that these organisms and every other organism needs to do their cellular work to stay alive. So it's a critically important process. Until the evolution of the process of photosynthesis, every organism that predated these autotrophic organisms were consumers. And you know that you can't have a self-sustaining planet if everybody's a consumer. You've got to have some energy producers, or more accurately, energy transformers as well, for the process to keep going on. And if we don't damage our planet too badly in other ways, photosynthesis in living organisms should continue as long as we're getting that input of solar energy, and these organisms can then convert that into the chemical energy of glucose, which can then be turned into ATP by cellular respiration. So again, remember that autotrophic organisms don't have just photosynthesis. All of these organisms have to have the ability to convert organic molecules into ATP by cellular respiration. But in addition to that, the autotrophic organisms have this unique special ability to trap the sunlight energy and turn it into the glucose, which is then used as a fuel in that cellular respiration process. Now, not all of the breakdown of glucose happens in the mitochondria. Okay, so we've got groups of organisms that extract energy from glucose by fermentation and by another process called anaerobic respiration we didn't get into. But the most efficient way for organisms to extract the energy from glucose is by aerobic cellular respiration in a mitochondria. So, life depends upon producers on this planet. Here are some slides of some organisms that are autotrophic or photosynthetic. We've got some ferns, some large algae, some little unicellular organisms. All of these are eukaryotes. If we're talking about the three-domain system of classification, then all of these photosynthetic organisms would be in the same domain. They are in different kingdoms. Plant kingdom. The taxonomy of algae is a little bit confusing sometimes. I guess more typically they're considered plants as well. But this would be the kingdom Protista. Remember that the other two kingdoms within the domain Eukarya are animals and fungi, and they're not photosynthetic. Okay? Unfortunately, we can't walk outside and soak up some rays and make our own glucose. So we're consumers. Down here, we've got two different types of bacteria. Here's one that you have seen before. These are prokaryotic filamentous anabina, and they belong in the domain Prokarya. And then we've also got some little tiny bacteria here that are the archaea bacteria, which are also photosynthetic. So you find representations of photosynthesis throughout all of the domains and in several of the kingdoms in those domains. So here's the process. Okay, we've got some reactants and we've got some products. This is a balanced equation, and what might be kind of confusing is that we see the same molecule both as a reactant and a product in photosynthesis. But these are not the same water molecules as these are. These are actually different water molecules that are formed. You can see that the major reactants here are inorganic molecules, carbon dioxide and water. These are molecules of low free energy. Okay. Some of the products of photosynthesis are also molecules of low free energy. But one important molecule right here, glucose, is a molecule of high free energy. Okay? So if we look overall at the free energy of the reactants and the free energy of the products, it looks like this. So overall, what kind of a process is photosynthesis? It's an endergonic process, right? It is an uphill set of reactions. Okay?
Those are
What's
kind
that?
of backwards.
Oh, okay. So the products are up here, the reactants are down here. Okay? We're going uphill. So what's driving this? Well, the energy of sunlight is driving this. Okay? And specifically, that sunlight energy is going to have to be converted into two forms of chemical energy to really drive those endergonic reactions. What are the two forms of chemical energy that are produced by sunlight energy? Well, ATP is produced. And then basically a cousin of the coenzyme NAD, NADP, is also reduced during photosynthesis. Okay? And then these forms of energy are used to convert CO2 and water into the glucose. The process of forming ATP has a lot of similarities to the way ATP was produced in aerobic cellular respiration. Remember that we said that in the mitochondria, this process of oxidative phosphorylation was the mechanism used to produce most of the ATP. 32 to 34 out of the 36 to 38 ATP. What does that mean, oxidative phosphorylation? It means that we're oxidizing organic molecules, removing the hydrogens, passing them through an electron transport chain, generating a hydrogen ion gradient, and using that hydrogen ion gradient to make ATP. So it is the oxidation of organic food molecules that drives oxidative phosphorylation. ATP production in photosynthesis occurs by the process of photophosphorylation, not oxidative phosphorylation. So sunlight energy is driving the synthesis of ATP, not the oxidation of food molecules. And that's kind of a subtle, but it's an important difference. Fortunately for you, the mechanism of both oxidative and photophosphorylation are very similar. And a lot of the things you learned about oxidative phosphorylation will apply to photophosphorylation as well. So it's not like a whole new process that you've got to relearn. Another thing about photosynthesis that's significant is that, like cellular respiration, this is an oxidation-reduction reaction. So what's happening in this particular process? Carbon dioxide is being reduced into glucose molecules, and water molecules are being oxidized into oxygen. So basically, we're stripping the hydrogens off of water, adding them onto carbon dioxide, and making a reduced organic molecule glucose. This is not how early studiers of photosynthesis thought things were happening. When people first started to understand what the reactants and products were of photosynthesis, they looked at that equation and said, well, I wonder what's going on here. What mechanically is going on to convert carbon dioxide into glucose? And what seemed to be most logical is that if you just add some carbon to H2O, what do you have? You have carbohydrate, right? CH2O. That's the empirical formula for glucose. So the early people who studied photosynthesis just kind of assumed that the way photosynthesis works is we split carbon dioxide and we break bonds. And when we take the carbon and add it to water, we form carbohydrate. And what's left? Oxygen. That's what seemed to be the logical mechanism of photosynthesis. And for a long time, everybody just assumed that's how it worked. But in science, what is logical isn't always correct. And so some experiments were done later on, particularly following World War II, when we started developing radioisotopes and could use those as tracer molecules to follow what happened, chemical reactions, where we started labeling, for example, water molecules with isotopes that we could detect. And all of a sudden we found out, hey, carbon dioxide's not what's being split in photosynthesis. Water molecules are actually being split. And so the evidence that we got from these experiments wasn't consistent with the hypothesis that everybody had kind of assumed was correct. So that's an important lesson to understand about science is just because something seems logical doesn't necessarily mean that's actually what's happening. So in photosynthesis, water molecules are being broken down. Bonds are being broken. Hydrogens from those water molecules are used to reduce CO2 into sugar, and oxygen is produced as a byproduct. Now, something to understand that's important. Actually splitting those bonds and breaking water molecules down into its atoms is not the same thing as dissociation. Okay, water molecules dissociating into their ions, is dissociation. This process of breaking the bonds, the covalent bonds, forming the atoms of oxygen and hydrogen is a different ballgame. And this breaking of bonds requires a fairly significant amount of energy. This happens on its own. Okay? So where's the sunlight coming from that breaks these bonds during photosynthesis? That's what the sunlight energy is doing, is it's actually breaking those bonds. When water molecules are split, the hydrogens don't immediately glom onto CO2 to make carbohydrate. Instead, they are grabbed by a coenzyme, and eventually the coenzyme will donate those hydrogens to CO2 to make the sugar. So there's this intermediate involved. So we have to account for that. As water molecules are split, a hydrogen ion gradient is formed. And that hydrogen ion gradient is used to make ATP. So these are some of the kind of things we're going to have to account for as we dig into this process of photosynthesis in a little bit more detail. Okay, so here's a overall summary of what's happening in photosynthesis. And right now we've got some kind of black boxes right here that we're just going to say something's happening here, but we're not quite ready to dig into the details of it yet. In general, the many reactions that make up photosynthesis fall into two general categories. The first category of reactions are reactions called light-dependent reactions. Sometimes these will just be referred to as the light reactions. Okay. The second category are the light-independent reactions. They used to be referred to as the dark reactions. And that was an unfortunate choice of terms, as it turned out. Because what does that imply? Well, light reactions imply you need sunlight for that to happen, and that's true. Dark reactions imply you have to have the absence of sunlight. That's not true. These can happen in either the light or the dark, given certain circumstances. So these are not dark dependent. So I prefer the term, and most books do now too, light-dependent reactions, light-independent reactions. In order for the sunlight energy to be harnessed and grabbed, it's got to be absorbed. And in order for light to be absorbed, something's got to be grabbing that light and kind of soaking it up. And the molecules that absorb the light in photosynthesis are called pigments. What's the primary pigment in photosynthesis? Primary pigment is chlorophyll. But we're going to see that there are actually several forms or types of chlorophyll. There are chlorophyll A's, there are chlorophyll B's, there are chlorophyll C's. There are some other chlorophyll, or some other pigments, that are referred to as accessory pigments. And the two primary accessory pigments are compounds called carotenes and xanthophylls. Now, this idea of a pigment doesn't just relate to photosynthesis. It relates to a lot of other things as well. Paints and dyes are pigments. What's a characteristic of a pigment? A pigment, because of its molecular structure, absorbs certain colors or wavelengths of light, but not necessarily all of them. Some of those colors or wavelengths of light might be reflected by a particular pigment. When we look at something that has a color, what is that color really telling us? It's telling us which wavelengths of light are reflected, are not absorbed. And they bounce off of the molecules in your fabric of clothes you're wearing or of a painting you're looking at. And those wavelengths of light aren't absorbed but are reflected and bounce back to your eye, stimulate rods and cones in your eye, and send electrical signals to a portion of the brain that gives us a perception of a color. So what color are chlorophylls? They're green, right? So what are they doing? They are absorbing the blue and the red, but they are reflecting the green color. In order for sunlight energy to do work of doing things like making ATP and reducing NADP, those wavelengths of light must be absorbed. So things that are reflected aren't really helpful in making photosynthesis happen. Did any of you ever, when you were little, do a science fair project where you grew plants under different colors of light? If you didn't, what could you expect would be the result if you grew them under green light, for example? Should they flourish and do very well? No. Because they're reflecting, their pigments are reflecting those colors of light. They're not absorbing them and turning them into forms of energy that can be used to do cellular work. You may know what color carotenes are? Orange, right? You know what color xanthophylls are? It's a little tougher one. They're yellowish pigments. Last fall, we could see that the leaves of plants on campus had a mixture of chlorophylls and then some of these accessory pigments. Typically, the chlorophylls mask out the presence of the other types of pigments. The green kind of predominates. But as chlorophyll breaks down, that's one of the less stable pigments. Then you can see some of the yellows and oranges and things left behind which represent the accessory pigments, which still haven't quite broken down. The organelle of photosynthesis is the chloroplast. And its structure is really well adapted to its particular function. We'll look at the structure in more detail later on, but one of the things that chloroplasts have is a series of membranes inside the chloroplast called thylakoids. And the membranes are where we find these pigments that are absorbing the sunlight. This is where these light-dependent processes of photosynthesis are going to take place. So they're associated with these membranes in the chloroplast. Additionally, the inside of the chloroplast is filled with a fluid-like material. It's kind of like the matrix of the mitochondria. It's got a bunch of enzymes, water. It's kind of like the cytoplasm of cells. And that fluid-like material is called the stroma. And that's where the light-independent processes are going to take place. So the chloroplast has kind of different parts to it where different processes of photosynthesis take place. It's not unlike the mitochondria. Okay, in the mitochondria, Krebs cycle or citric acid cycle reactions take place in the matrix. The electron transport chain takes place in the inner membrane. Okay, so in the chloroplast, thylakoids are the light-dependent reactions. Stroma are the light-independent reactions. So, on this particular little diagram right here, what's going on? These three boxes right up here, which include light absorption, electron transport, and chemiosmosis, are all light-dependent phenomena. So these are going to be associated with the membranes of the chloroplast. This process down here, which is called carbon fixation, is the light-independent process. And that happens in the stroma. So CO2 fixation. Another name for that is the Calvin cycle. So thylakoid membranes and stroma, two different locations within the chloroplast. Okay, so what's going on in the light-independent process? What's happening in the membranes? Well, a pigment like chlorophyll is going to have to absorb that light energy. Anything that it reflects doesn't help. So the blues and the reds, particularly, of that spectrum of visible light are what kind of drive photosynthesis. What happens when light's absorbed? Well, an important thing happens, and that is water molecules are broken apart. Bonds are actually broken. Covalent bonds are broken. And hydrogen atoms and oxygen atoms are released. And if a couple of those oxygen atoms are in vicinity with one another, they're going to bond to one another. Why? Because an oxygen atom is unstable. Okay, it's got an incomplete outer electron shell. It's got its inner 2 electrons in the first shell, and then it's got 6 in the next shell. It needs 2 more to be stable. So what's it do? It finds another oxygen atom and binds to it. Now they're happy, now they're stable. What do we have then? we have a molecule of oxygen. That's a waste product. Plant cells are going to get rid of that waste product. As it builds up to a higher and higher concentration, it's going to move by diffusion out of the plant cell into the atmosphere. And what's it going to do? It's going to change the atmosphere one molecule at a time. Plants pollute their atmosphere with oxygen. Prior to the evolution of photosynthesis, there wasn't free oxygen in the atmosphere. The atmosphere was dominated by things like ammonia, a lot of nitrogen, which we still have, but no free oxygen. So prior to the evolution of photosynthesis, was aerobic respiration possible? No oxygen. Okay. Also, I mentioned this, when oxygen started to accumulate in the atmosphere, oxygen atoms could react with one another to form ozone. And what did that do? That filtered out some of that harmful cosmic radiation, which was just frying things on the surface of the planet. So everything had to live in the water because water can filtrate that harmful cosmic radiation. Now organisms could transition to the surface of the land when this protective ozone layer developed. But that needed oxygen, which was produced by photosynthesis. So look at how the evolution of photosynthesis changed our planet and the dramatic changes that occurred within it. Well, something else interesting happens during absorption of light. When these pigment molecules start absorbing the light energy, electron transport happens. Electrons start flowing in that membrane. Now you might look at that and say, how does that happen? That seems kind of unrealistic. Well, you see examples of that all the time in the real world where light can cause electrons to start flowing. If you have a solar-powered device, like a calculator, you flip it open, there's a solar cell that absorbs the light energy, and the absorption of those photons of light energy causes electrons to start moving. What's moving electrons? It's a current. That powers your calculator. In photosynthesis, the light absorption starts moving electrons through an electron transport chain. And what happens when electrons start moving? We can form hydrogen ion gradients. And we can use that through the process of chemiosmosis to make ATP. Some of those electrons are used to reduce a coenzyme. Now that's kind of opposite to what happens in cellular respiration. In cellular respiration, reduced coenzymes like NADH and FADH2 dump their electrons onto an electron transport chain. In photosynthesis, electrons are pulled out of the electron transport chain and put onto these coenzymes. So it's almost kind of like reverse electron transport. Now, one thing you got to think about is, where do those electrons come from? Because if we pull them out of the electron transport chain, we need to stick more electrons in to replace those. Where do they come from? So we'll talk about that. So here are the main events of the light-dependent reaction of photosynthesis. Light's got to be absorbed. If that doesn't happen, this doesn't happen at all. When light's absorbed, water molecules are split. Oxygen is a waste product. Some of the hydrogen is eventually transported to NADP, reducing it. That's potential energy now. And as the electrons move, we can make a hydrogen ion gradient to make ATP. So these are the three major products of the light-dependent reaction. The reactants are water and light. Any CO2 involved yet? Nope. No involvement of carbon dioxide so far. In the light-independent reactions, carbon dioxide is going to become reduced into carbohydrate. It That requires some hydrogen, right? Where does the hydrogen come from? This coenzyme. What does ripping the hydrogen off of this coenzyme do? Recycles it. Turns it back to the oxidized state again so it can keep grabbing more hydrogens. This particular process is endergonic. CO2 is down here. Glucose is up here. What drives that? The hydrolysis of the ATP produced in that light-dependent reaction. So do you see how these two are interrelated to one another? What does the light-independent reaction require? The products of the light-dependent reaction. So as long as the sun is shining and producing these, if there's CO2 present, glucose can be formed. Could this potentially happen in the dark? Yes, if we stockpile some of this stuff in the daytime. So if there's still some of this left over when the sun stops shining, CO2 can still be fixed into carbohydrate using these energy sources. So this is light independent, and this is light dependent. So that's the big picture. Okay? Now what we need to do is kind of dive into some of these little black boxes a little bit and kind of see what's going on. As I said, fortunately, these kind of black boxes are very similar with some nuances to what we saw in cellular respiration, particularly this one, chemiosmosis, very similar. This one, there's a little bit of variation and wrinkles between electron transport and cellular respiration and photosynthesis. So let's start here. Let's start with this light absorption phenomena first. So here's our chloroplast. Chloroplast has an outer membrane, and then it has a series of inner membranes which are stacked up, kind of like stacks of coins. What's that stacking a adaptation to? Increasing surface area, okay? Because what's associated with those membranes? The pigment molecules. The more surface area you have, the more pigment you can have. Light energy is driving these light-dependent reactions, and water molecules are needed to make these light-dependent reactions occur. Now let's see if I've got a slide that shows this. One of the things that's important to see about these stacks of molecules is that the membrane has a space inside of it. Okay? So in photosynthesis, we have these thylakoid membranes, and there's a space inside the membrane, and then these thylakoids are floating around in this fluid-like stroma. So what do we have? We have two compartments, the stroma and the space inside the thylakoid, separated by a membrane. What's the significance of that? In order to have chemiosmosis occur, we need a membrane separating two spaces, just like we did in the mitochondria. So in the light-dependent reactions, water molecules are split. As light is absorbed, oxygen is produced. And because electron transport occurs, we're producing this reduced NADP. That's where the hydrogen goes from the water, here. And we make ATP by the process of chemiosmosis. Then that's used in the stroma, in the Calvin cycle, to reduce CO2 into sugar. So we'll continue on here tomorrow.